as a change. So I'm Livia. I'm a research fellow. I work with Sarah Jane Blakemore in psychology, and I'll speak today about multivariate methods in fMRI to study within subject changes in brain signals. So just a very brief introduction to pattern-based approaches, as I'm guessing most of you are familiar with them anyway. So in general, we know that the spatial patterning in human fMRI signals contains information about a person's mental state. And the advantage of using pattern-based approaches is that they do have a higher sensitivity typically compared to univariate fMRI approaches like the classic GLM to detect differences between conditions. So one way then to kind of take advantage of this higher sensitivity is to study how in the same person, for example, I'm going to show stress, but perhaps other aversive states change brain states. So I will talk about two different methods, which is representational similarity analysis and MVPA. And I won't talk much about the details of it because they are both described really well in the literature. But what I'll try to do is give examples of how we've been using these methods to actually address these questions. And yeah. So the first study I want to introduce is a study where we studied how social threat modulates reward processing. So for that, we had participants play a lottery task, and so they were playing a lottery where they were deciding whether they wanted to, on each trial, to play a specific lottery that had a different winning probability. And they played this task either for themselves or for another person. And in this study, we're specifically interested how stress would affect reward representations or value representations for oneself and for another person. So we asked participants, so the same people, to undergo the lottery task either under social threat. So we induced stress in the scanner using a protocol where participants have to undergo mathematical calculations and are being watched doing that, which works quite well to stress people, and then in a control condition. And we were specifically interested in the MPFC as it has been most directly been associated with reward representations for value-based decision-making. And the way we did this was that we used the functional localizer. So here we had a different task that was independent, where participants were given rewarding stimuli, and we looked at the activation in each person within the MPFC and extracted the individual regions of interest based on the function of each person. So then, in order to extract the patterns for the RSA, we extracted from each of our ROIs the pattern response, and then we compared those. So we calculated pairwise distances between each of our conditions of interest, and the distance was 1 minus correlation. So we had 4 conditions that we were interested in, either a high reward task that was relevant to the self, so participants were able to win money for themselves, or a low reward, so that just means less money, or those 2 conditions for another person. And we specifically looked at the representations between high reward self and high reward other, and high reward other and low reward other, because we wanted to look at how stress modulates representation for other people. And we had 2 specific hypotheses that we wanted to test against each other. So one hypothesis was, well, perhaps stress increases the similarity between high reward for self and other, or in other words, it might inhibit the self-other distinction. So you could imagine once we're stressed, perhaps we're not really well representing who the reward is for, and that's why we might act more prosocially. So I should add, we wanted to test a behavioral finding that has been reported in the literature, that people sometimes behave more prosocially when they're stressed. So we wanted to see whether we can find kind of a neural correlate for that. Well, the alternative hypothesis was that perhaps stress actually actively shifts the focus to another person, where we would predict that the representation of high reward and low reward would become larger or more dissimilar. So participants would track the magnitude of reward more for the other person. And what we actually found was the evidence for our second hypothesis. So stress increased the dissimilarity of high rewards and low rewards for others, and that it was also correlated with how favorably those people played for another person. So this is one example how we use this kind of method to ask a specific question about how stress can affect brain signals. And this was kind of taken then as, or supports this idea that there is kind of a third stress response, which is called tend and befriend, which means that other than fight or flight, people also tend to show this increased affiliation to others as a stress response in order to seek kind of support from others. And this was evidence for it on a neural basis. So the second study I wanted to introduce is a study where we looked at neural correlates of the desire to engage in social interactions. So here we isolated participants. So participants spent 10 hours without social interactions, which led to increased social craving and loneliness. And then we were interested in the representation of the desire to engage in social interactions. And to compare this to something that we kind of already were quite sure we would know how that would look like, we had a control condition that was food fasting. So we asked the same participants to not eat for 10 hours, and then we also they also underwent fMRI scanning, and we were specifically interested in activation patterns in the substantia nigra, which is known to represent motivational signals and cravings. So we specifically looked only at this, or we looked at others, but our target region was the substantia nigra. So just to quickly talk about univariate activity, what we found here was, well, first of all, that the activity within, so the average activity within the SNROI was increased when participants were viewing pictures of food after they had been fasted compared to after they had been isolated. And that was correlated with their self-reported wanting of food. So those people who wanted less food also showed less activity in the SN. And we found a quite similar response in the SN after isolation. So after isolation, participants showed stronger activity to social images compared to fasting. And again, this was correlated with their self-reported wanting of social contact. So then we were interested in, this looks like a quite similar response, but the univariate analysis didn't quite allow us to test for that. So we were interested, well, how similar are those neural signals actually? So in order to do that, we used MBPA and specifically linear support vector machine classifier. So first of all, we wanted to test whether we were actually able to extract any meaningful pattern activation from the SN, as it's kind of a very basic sort of brain region. We were not sure if there was meaningful information in the spatial pattern of the signals. So in order to do that, we first trained the classifier using a six-fold cross-classification method where we left one test set out and then averaged across all 6 folds. And essentially, we tested we trained the classifier on this data, and then we also did a permutation data set where we generated a null distribution where we randomly permutated the labels and essentially let the classifier train on meaningless data. So then we tested whether the actual real classifier would be significantly better than the one that had been trained on nonsense data, and we found that yes. So we had within the pattern of information in the SN, there was meaningful pattern of whether someone looked at food or social pictures. So this was all done only for the fasting condition, as it was mainly just to look at whether we would have any signal there. So what we then did was that we actually wanted to cross-classify across motivational states. So we also had a baseline condition where participants were neither isolated nor fasted. And the way we did this was that we trained the classifier on all of the data that we had in the fasting condition to between neural patterns of a hungry person looking at either pictures of food or control pictures. And then we tested the classifier on 2 different data sets, which were both a person looking at social pictures versus control pictures, either after experiencing isolation or after no isolation. So the idea was the visual input in the testing data set would be different because now they're looking at social pictures. But in one condition, the motivational state perhaps is similar to that one because in both they would be have an experience of deprivation, while here they would not. So what we found was actually exactly that our classifier was able to, above chance, distinguish neural patterns of whether this person had been looking at social pictures versus control pictures, but not in a baseline condition. So that suggests that there is a shared neural signature between the neural patterns of hunger and loneliness. And to test the kind of similarity in those patterns more closely, we then again used RSA analysis and again pairwise representational distances. We were interested in whether the pattern within the SN would be more similar or wanted to quantify the similarity between hunger and isolation. So we looked at the pairwise distances between the condition where a hungry person was looking at food or the same person not hungry looking at food. So now the visual input is the same, but the motivational state is different. And then we looked at the representations when the hungry person was looking at food and a lonely person was looking at social pictures. So now the visual input is different, but the motivational state perhaps is similar. And then we generated a ceiling condition where there was a maximally dissimilar. So here, neither the visual input nor the motivational state was the same. And then we tested those two against the ceiling. And what we found was that the motivational state was significantly different from ceiling, but not the stimulus. So that means that the neural patterns became more similar when the motivational state was the same or a similar one rather than the stimulus. So again, that suggests that the deprivation alters the neural patterns to become more similar, regardless of whether the deprivation is social contact or food. All right, so that has led kind of to support this idea that loneliness perhaps is a motivational drive rather than just a aversive emotion without any redeeming kind of purpose. But the idea is that perhaps loneliness is very similar to hunger, that a person that is lonely is motivated to seek out social contact in order to decrease loneliness, just like someone who's hungry would seek out food. All right, that's it for me. Thank you all.
Thank you so much, Olivia.